Thank you so much. That was really wonderful information. And now that we know you're here, Dr. Shapar, we're going to send lots of chordoma patients your way. Yeah. So we're going to move now to chordoma research updates. We're going to start off with Josh Sommer, who, as you likely all know, is co-founder and executive director of the Chordoma Foundation. Josh is now an 18-year survivor of chordoma and continues to push chordoma research and the Chordoma Foundation to new heights and ever greater accomplishments. And Josh will share more about exciting research that's underway. Welcome, Josh.
Well, thanks, Shannon. And while she's getting my talk up, thanks to all of you for being here. It's really a highlight of the year to get to bump into old friends and to meet many of you for the first time. So really appreciate you taking the time. So a lot of the day has been focused on living with chordoma. And we're going to shift gears now in the next part of the day to talking about improvements in treatment and research that's leading to new therapies. I'm going to talk about some of the recent research advances at the Chordoma Foundation and some of the themes that we're pursuing in our research efforts. And then following my talk, Dr. Gandhi and Dr. Walensky from Northwestern, as well as Dr. Ariada, if I'm saying that correctly, are going to present on some of the latest treatment advances from a radiation standpoint, from a surgery standpoint, as well as some of the cutting-edge research that's going on at Northwestern. So the way we're going to do this is each of us will talk for, I think, about 30 minutes or so. We'll save questions for the end, and then we'll have a panel discussion and kind of all take Q&A together. Without further ado, many of you are probably familiar with this trajectory, but for those of you who I'm meeting for the first time, just a quick background, kind of quick snapshot of my background. I was 18 years old when I was diagnosed with chordoma back in 2006. I had a skull-based tumor that was diagnosed because I had persistent headaches and went in to get an MRI, which I think probably no surprise to all of you, revealed the kind of shocking discovery of a tumor right in the very center of my head. I was fortunate to have a great surgery, a complete resection, and did great for about 16 years before, on a routine MRI, finding out that I had a recurrent tumor very close to the initial tumor in my skull base. I was very lucky because when I was initially diagnosed, I was under the impression, and I think it had been conventional wisdom, that a recurrence is really bad news and not always operable, et cetera. But I was very fortunate. It was caught relatively early. It was small, and it did happen to be operable, and I had a successful surgery. And case in point, I'm here today feeling great and exceedingly grateful. Shortly after my most recent surgery in 2022, we welcomed our second child, who's now almost 2 years old. And I feel so grateful. I think all of us have heard the average survival of being 7 or 8 years, and you obviously never know what cards you're going to get dealt or what the future is going to hold, but being here today with all of you is just something that I never imagined would have been possible, and I feel so tremendously grateful for. I would say one of the other silver linings of this journey or ordeal has been being a part of this community. I think none of us asked to be a part of this community. This is certainly a club that probably none of us wanted to join, but nonetheless, I think being part of it for me has offered or provided a source of strength. I think it's really true that there is power or strength in numbers. Certainly from a practical standpoint, when I was first diagnosed and going through my recurrence, the support and guidance that I got from so many in this community was a tremendous practical help. But also I really find strength and hope in realizing that together we have the power to do what none of us can do individually in terms of changing the outlook for this disease. And that's really what the Chordoma Foundation is all about. It's a vessel, really, for all of us who are affected by this disease to pool our efforts, to pool our resources, to bring about a better future for all of us affected by this disease. So a brief overview about the Chordoma Foundation, and I know many of you already know this, but for those of you who are new, basically we have, our mission is to improve the lives of those affected by chordoma and to lead the search for a cure. And to achieve that, there's 3 goals that we're pursuing: better treatment, better care, and a better patient experience. I'm going to talk just very briefly about each of these areas and then go deeper into research. So first focusing on our patient services aimed at helping to improve the experience of those of us affected by this disease. As you've heard from Shannon and Andrea and Danny and others, over the years, Shannon really, with Shannon's leadership, we've built out quite a robust and diverse suite of resources and programs to help address the myriad issues and challenges that those of us who are affected by this disease face at all stages of the disease, whether it be initial diagnosis, whether it be a recurrence, and everything in between. So I won't touch on all of these, but I think just suffice it to say that the foundation, on a daily basis, I think where we see our the most important role that we play is being there as a source of support to address any need that might arise. So I would just encourage everyone here, if there is ever anything that you need, a challenge that you're facing, to not hesitate to reach out. Or if there's things that you feel like would be helpful that you're not getting from the Chordoma Foundation, we're always eager to improve. The second pillar of the foundation is what we call healthcare improvement, working to level up the care that is provided to patients by clinicians across the country. And we do this in a couple of different ways. Both Dr. Walensky and Dr. Gandhi have participated in our research workshops. So we bring together researchers and clinicians from around the world to exchange the latest knowledge, to exchange research updates. Additionally, we've organized a number of meetings of clinicians from around the world to really try to drill down and reach clarity on what are evidence-based best practices for the treatment of this disease. I'm sure it's maybe not a surprise, given that so many of us get differing opinions from different doctors that we speak with, that there is a wide variety of opinions out there. There's a lot of variation in care. Thankfully, I think that is improving, and I would argue that that's in part thanks to the convening that has happened over the last 15 years or so, as well as the treatment guidelines that have resulted from that. Admittedly, this is, I would say, the most underdeveloped aspect of the Foundation's mission, and an area that we see significant potential to enhance or continue to expand and invest in further. Now, where I'm going to focus the lion's share of the rest of the talk is on research. And there's a lot of research that needs to be done. There's research that needs to be done to understand the basis of the disease. How does chordoma arise? There's research that needs to be done to identify biomarkers that might predict which patients are going to do well and which patients are going to do less well, which patients are going to benefit from radiation or systemic therapies, et cetera. There's a wide variety of questions, and at our research workshops, we try to collate these questions. However, you have to focus. And the number one thing that is clear is missing from the treatment of chordoma is effective systemic therapies. I think, as you'll hear from Dr. Gandhi and Dr. Walensky, and probably as many of you have experienced firsthand, surgery and radiation have become amazingly advanced. Like, I am in complete awe of what surgeons and radiation oncologists are able to accomplish in treating this disease. And in many cases, it can be curative, or it can allow patients to have a good quality of life for a long period of time. But the problem is, in a substantial fraction of chordoma patients, even despite how great surgery and radiation have become, a lot of patients will recur and eventually progress beyond the point of being able to be controlled by surgery and radiation. And when that happens, that has historically been where the biggest gap has been. And so the lion's share of our research focus is to try to address that problem. So when we think about developing systemic therapies or drug therapies, what's the purpose? And the reality is there's actually multiple purposes. They might play different roles in the care of different patients, and this will probably change over time. As is the case for many oncology drugs, the primary context in which they are developed is for patients with advanced disease. And so that's initially the main focus, and where drugs are being tested is for patients who have already failed surgery, who have already failed radiation, to try to provide a better option than what exists now. But that's not where this ends. That's the kind of a stepping stone towards the ultimate goal of having systemic therapies that could be offered with surgery and radiation to perhaps reduce the risk or prevent recurrence, or maybe even eventually be given instead of surgery or radiation. Not that we want to put the surgeons, radiation oncologists out of business, but especially in cases where surgery or radiation would be highly risky or morbid, could we develop a systemic therapy that is safe and effective enough to serve as an alternative, a better alternative to surgery and radiation? So that's where this is heading. That's the ultimate vision. I think we also know, and I think this is important context, so there's very unlikely to be a silver or a magic bullet. There are very, very few instances of cancers that are cured with a single drug. Cancer is a heterogeneous disease. Not every tumor is the same. There's a lot of differences we know this now, there's a lot of differences within chordoma. And furthermore, chordoma, and as is the case for all cancers, is constantly evolving. And so what that means is that we can't put all our eggs in one basket. We can't say, we have great confidence that this is the one therapy that we should put all of our effort behind. We know that we're going to need multiple shots on goal, and ultimately a toolbox of multiple drugs that could be used in combination, that could be personalized to deliver the right drug to the right patient based upon the makeup of their tumor, and potentially that might be used in sequence if a tumor develops resistance to one of those drugs. So then the question becomes, where are these drugs going to come from? What is the path to these drugs going to be? And at a very, very high level, there's basically 3 different paths to these drugs. So the low-hanging fruit, the very easy path, is to try to repurpose something that already exists. There's roughly 3,000 drugs that are on the market that are approved by the FDA or European regulators that could be prescribed basically tomorrow. 250 roughly of those are approved for cancer. So that's the near-term possibility. And the great thing there is that the billions of dollars that it takes to develop drugs have already been invested, and all we're doing is trying to see if any of those could apply to chordoma. So that's the obvious, the low-hanging fruit, the place to start. I'll talk more about that in a moment. The other option is applying experimental drugs. So at any given time, there's roughly 7,000 drugs that are in development. This is for all different diseases, and of those, there's roughly 1,000 that are in development for cancer. And this spans the gamut from targeted therapies to immune therapies, et cetera. So there's a huge amount of investment. And for the most part, it's hard to make a business case to invest in a drug for chordoma. However, these drugs have already had the lion's share of the initial investment committed based upon their potential for other diseases. And if we can apply them to chordoma, there might be a window of opportunity where actually there is a financial incentive for a company to invest in chordoma, because there's substantial incentives that are provided to companies to develop drugs for rare diseases. And I won't go into great detail there, but there's this window of opportunity when drugs are in phase 1 trials. They haven't yet mapped out or kind of set in stone their clinical development pathway. And if we could convince a company that chordoma is the appropriate path or the best path to getting their drug approved, that actually can be a very substantial business case for investing in clinical trials for this disease. it also has the advantage of, like repurposing, these drugs are available now in the clinic. We don't have to wait many years or decades before they're even available. The key thing is that that window of opportunity is oftentimes narrow. It's maybe only a couple years when there's a clinical trial that might be open to a chordoma patient. The third path is developing new drugs. This is clearly the longest, the hardest, the most expensive. It's been well studied that developing a new drug, costs well over $1 billion on average. And so this is a big undertaking, and not something to be taken lightly, and probably not something that can be done a lot of times. However, if the biology leads us in that direction, it must be done. And so I'll talk a little bit about some efforts underway to develop new drugs for chordoma, because we do think there's a compelling biological reason to take that on. So we don't yet know which of these drugs are going to be the ones that work for chordoma. And likely we're going to need to pursue multiple of them in parallel. And that's going to need to be guided by the biology of the disease. What we can know with some degree of certainty is that regardless of which drugs ultimately end up providing the answer, that the path to identify them and bring them to chordoma patients is going to traverse basically a predictable kind of set of research areas. So ultimately, they're going to culminate in clinical trials. To enable those clinical trials, you're going to have to do preclinical research. That means study those drugs in cells and animals. Maybe you're going to have to do drug discovery if the drug doesn't already exist. That all is going to be determined by understanding the biology of the disease and identifying potential therapeutic targets. So that tells us we have to do a lot to study the biology and understand kind of all the different layers of what's going on, peel back the layers of the onion basically to figure out what targets to go after. And all of that research, all of that target discovery or basic research is all dependent upon having a set of resources that researchers need to be able to study the disease. So progress in each of these areas of research is going to be needed to enable us to ultimately achieve the promise and what we know is possible of better treatments for this disease. So that is what is kind of incumbent upon us as a foundation and as a community to ensure that progress can be made at each of these stages and is being made at each of those stages, and not leave that to chance. So often we see for different diseases, money will be raised, and there's some great, interesting question that is worthy of being pursued and needs to be answered, and money is invested in that particular project. But without the full context, without thinking about this entire end-to-end process that is and everything that's going to be required to ultimately bring whatever comes out of those studies to fruition in the form of therapies for patients. And that, I think, is kind of a defining feature of the Foundation's research strategy, is this end-to-end approach, recognizing that progress at every step on this path is necessary but not sufficient, and we have to grab the bull by the horns and make sure that every step from A to Z happens. So how are we doing that? A number of different efforts. As I said, there's a whole bunch of things that are necessary but not sufficient. So it's not just enough to raise money and give grants, although that is very important. We need to create venues for knowledge exchange between researchers and venues for collaboration. So we do research workshops to enable that. We've built repositories of tumor tissue, of cell lines, of animal models, and thank you to all of you who have contributed tumor tissue to our biobank. This is an incredibly valuable resource for the research community. With those resources now in place, we've been ramping up in recent years our investment in research. So this means basically, for the most part, grants to academic investigators, including here at Northwestern. And we're very, very proud of the work and just thrilled with the work that's going on here, in particular in the lab of Dr. Catalina Li Cheng. We, as much as possible, try to stretch our dollars, and so we look for opportunities to partner with other foundations. And in this particular case, we have partnered with an immunotherapy-focused foundation called the Cancer Research Institute. So I think for 5 years now, we've had a partnership where we've co-funded immunotherapy-focused projects with this other foundation. But we fund research in a number of different areas. Additionally, as many of you may have heard, we've recently started a lab that is 100% dedicated to chordoma. And And why might we need our own lab? Well, because there's a certain set of capabilities that just didn't exist anywhere else in the world that were needed, actually needed by lots of different labs. And it would have been very difficult, costly, inefficient for lots of labs to try to recreate the same capabilities. So we've created them in a centralized lab and make those capabilities available to lots of different investigators and, importantly, to companies as well who are interested in the disease. And our lab focuses on two things. First of all, being a partner to investigators and companies to basically support their research and enable them to pursue hypotheses that they wouldn't be able to pursue on their own. And secondly, to proactively drive forward research of our own that isn't being pursued within academia or industry and for whatever reason doesn't fit within the kind of academic or industry incentive structures, but is nonetheless very important. Finally, we focus a lot on trying to enable patient participation in research, and that includes clinical trials as well as contributing to the natural history studies, et cetera. So there's a lot to talk about here. I'm going to try to condense it into kind of just a very high-level snapshot of where we've come over the last 16, 17 years together. So when the foundation started, we basically had no research-enabling resources. There was no centralized source of tumor tissue. There was one valid chordoma cell line. There were no animal models. And now we have abundant preclinical models, abundant tumor tissue. This is not a problem that is fully solved, but increasingly it's in the rearview mirror. Additionally, 15, 17 years ago, there was almost nothing known about the biology of the disease. And because of the work of literally dozens of labs across the world and because of the philanthropic support of many of you in this room, the field has come a really long way in terms of understanding the biology of the disease, identifying a number of different therapeutic targets, some of which point to repurposing opportunities, and one of which points to a very compelling opportunity to develop new drugs, and that is the protein brachyury. Probably this is a term that many of you have heard of. It's used as a diagnostic marker for chordoma, but there's also been an accumulation of multiple layers of evidence suggesting that brachyury is the most important gene that chordoma depends upon for its survival. And it is pretty much universally expressed in chordoma, and chordomas appear to be universally dependent on it. So basically that tells us that while repurposing opportunities are interesting and promising, in many ways, a lot is suggesting that the holy grail is developing drugs against brachyury. So the problem is brachyury. So quick sidebar here. When you hear brachyury, it actually refers to two different things. Brachyury is a gene, so it's a stretch of DNA that is in every cell of your body. That gene encodes for a protein. So the gene basically is like the blueprint, and the protein is where the action is. So when pathologist looks at brachyury in a chordoma cell, what they're looking at is the protein. When we talk about targeting brachyury with a drug, we're talking about targeting the protein. The problem with the brachyury protein from a drug discovery or development standpoint is that it doesn't have features on it, pockets, crevices, et cetera, that are easy for drugs to interact with, for chemical compounds to interact with. And historically, as a result, it's been considered undruggable. However, I think this is actually one of the perhaps most encouraging themes that have run throughout the last 15, 17 years. We can never predict what technologies are going to come next. We can never predict what breakthroughs are going to come next. If we could, it would have already happened. And so, early on in the days of the Chordoma Foundation, we kind of had hints that brachyury was important. But at that point, everyone that we talked to said, don't waste your time. It's an undruggable target. You're never going to be able to drug this type of protein. Well, in the last 10 years or so, there's been a flurry of new technological breakthroughs that have made this type of protein, or made drugging this type of protein at least theoretically feasible. Now, this is not yet translated into drugs that are in the clinic, and we're determined to make brachyury, kind of put brachyury at the front of the line to have those different therapeutic modalities applied to it. So there's, at this point now, more than a half dozen drug discovery efforts focusing on brachy, applying these various therapeutic modalities, some of which we've supported in academic labs, some of which are happening in companies, some of which are happening in CF labs as well. On the translational research front, this is largely focused on repurposing at this point. There were 0 drugs that had ever been tested in the lab. 0 drugs that had ever been tested in the lab. So, when people talk about there not being effective drugs for chordoma, it's not because they don't exist, it's because they've never been tested. And that was largely because we didn't know which drugs to test and we didn't have the models to test them in. That's changed now over the last 15 years. So more than 80 drugs have been tested in mouse models through the Chordoma Foundation's drug screening program. Every single approved drug, not only in the United States, but in Europe and Asia as well, have been tested in chordoma cells. So we're answering the question systematically, is there any low-hanging fruit? Are there any approved drugs that could be effective for this disease? And of those efforts, there have been about 10 that have shown a great deal of promise preclinically. When the foundation started, there had been 1 clinical trial that was ever completed. In the time since, there have been quite a number, including 11 that the Chordoma Foundation has helped to initiate, and 5 more right now that are in planning. So what does that look like? Here you can see what plotted all of the clinical trials, all of the phase 2 clinical trials with systemic therapies for chordoma that have ever been published. Chordoma Foundation was started in 2007. So here there was this 1 clinical trial, I can't see my pointer, 1 clinical trial published in 2005. Between 2010 and 2020, there were a handful of clinical trials that were, I would say, opportunistic. None of these were based upon the scientific advances that I described in the previous slide. They were essentially kind of taking educated guesses and again, being opportunistic. New targeted therapies had been approved or discovered. Maybe they were being tested in one sarcoma or another type of cancer, and an oncologist said, let's try it in chordoma. But not because there was really any strong rationale or preclinical evidence. However, in the last year, we're now starting to see everything that I described in terms of the research advances in the lab bearing fruit in the clinic. So for the first time, there basically are 3 drug repurposing successes, i.e. drugs that were identified through laboratory research that have now completed clinical trials and demonstrated at least modest efficacy in chordoma patients. Now, clearly, 10% efficacy, so 10% of patients having significant tumor shrinkage is not good enough, but it's a toehold and it's something to build on. And I should also mention that one of these drugs, afatinib, which is maybe a drug that some of you have heard of, so the patients who responded on that drug, it wasn't a temporary response. The average duration of response was 28 months. So these are patients whose tumors were growing, then they shrunk, and then they stabilized for more than 2 years, which is really compelling. So what does this mean for patients? Well, now we basically have 3 drugs with response rates of roughly 10%. That presents an opportunity for combinations. Maybe that can be improved with combinations, and as well as sequential therapy. We've got to update the treatment guidelines so that insurance will start to cover these drugs. Biomarkers are needed to determine which patients are most likely to benefit from each of these drugs. But I think the thing to underline here is that this isn't good enough. There's a lot more progress that needs to be made. But I just want to mention that the benefit here is not something that is intangible. It's very real, and I think some of you might know some individuals who have benefited from some of these drugs, or maybe are benefiting from some of these drugs yourself. So these are friends of the foundation, people who have come to our patient conferences in the past who, all 3 of these gentlemen have been on afatinib and had long-term responses on afatinib. So it's just incredibly gratifying. And I recall early in the day, Mike talking about how, for many of us, there is this assumption that the work that's being done right now in the lab and the research that's being done might one day benefit people in the future. But it doesn't have to be that way. So much of the groundwork has been laid to enable rapid progress. And we're seeing now that individuals who have been a part of the Chordoma Foundation, who have contributed to our mission in significant ways over the years, are themselves now beneficiaries of some of the therapies that have come out of the research. So I take great hope in that, or I find great hope in that this is not just about benefiting people in the future, but there is actually a real chance to bend the course of the disease for each of us in this room. From a research perspective, this is important. It provides proof of principle that what we see in the lab does actually translate into the clinic. As I said, it provides a kind of an opportunity for combination therapies, and there is, in fact, right now, a clinical trial that is about to get underway that we're supporting in California, testing two of these drugs together, pembrolizumab and pemetrexed. It also highlights the need to better understand the kind of range of biology of this disease so we can select which patients or guide patients to the appropriate therapy. So there's about a half dozen projects that we're supporting right now to try to do just that, that will ultimately lead us to more personalized therapy. But I think the crux here is that we're at a turning point now. The low-hanging fruit, for the most part, has been mined. And I think to extend that analogy a little bit, there is a lot of evidence to suggest that the sweeter fruit is on the higher branches. We've learned a lot about new potential therapeutic targets that point to promising drugs that are being developed right now, not yet approved, but being developed, as well as I alluded earlier, to brachyury. So I want to talk now about some of this is all relatively new, some of the recent successes, or a big recent success we've had in applying an experimental drug to chordoma. So not an approved drug, but an experimental drug. So coming out of work that we supported at the Broad Institute of Harvard and MIT over the last 6-7 years, there was an effort to try to systematically understand every single gene that chordoma depends upon for its survival. And as I mentioned, brachyury is clearly at the top of that list, but it's not the only gene. There's about more than a dozen what they call dependencies or vulnerabilities. These are things that chordoma depends upon for survival that normal cells don't. And one of the targets that this group at the Broad Institute clued in on was this gene called SHP2. And the convenient thing, or the promising thing about SHP2, is that right around the time that this paper was, or this research project was maturing, there were a handful of companies that had been for years investing in developing SHP2 inhibitors, and they're all in a race with one another. And all of them started clinical trials right around the same time, around kind of 2020, 2021, 2022. And so recognizing, kind of seeing that window of opportunity on the horizon, we quickly tested a number of SHP2 inhibitors through CF Labs in models of chordoma. And it turned out that here you can see this, I guess you can't see my cursor, but in the top right, the black lines are tumor volumes of mice that are untreated, and the blue lines are tumor volumes of mice that are treated with SHP2 inhibitors. So significant tumor growth inhibition. Based upon that, so we shared that data with our medical advisory board, and very quickly members of our medical advisory board then enrolled chordoma patients on these, a couple chordoma patients on these open phase 1 SHP2 inhibitor trials. So the way we think of this basically is like there was this wave that had nothing to do with chordoma. That this was a target that kind of whose time had come in oncology. Lots of money had been invested in lots of different companies. And there was this window of opportunity, probably a couple-year window of opportunity when phase 1 SHP2 inhibitor trials were open to all comers. So it didn't matter that that chordoma is a rare cancer and that companies aren't intrinsically interested in it. Chordoma patients could get on these trials. And so a key role of CF Labs is to kind of be the jet ski here in this metaphor to kind of bring chordoma make chordoma able to take advantage of this window of opportunity, of this coming wave that only exists for a narrow window of time, by quickly validating targets that are identified by our academic collaborators and generating the preclinical data that can convince investigators as well as companies to enroll chordoma patients in trials. So the outcome of this was one of these trials actually enrolled 13 chordoma patients. This data was just published here in Chicago in June at the largest clinical oncology research meeting. And the results were pretty stunning, pretty remarkable. Tumors did not melt away. One patient had what would be considered a quite significant response. But what was really interesting is that almost every patient had some minor response. And that's something that's really not been reported before for most other drugs that have been tested in chordoma. And importantly, the duration of response for these patients whose tumors have shrunk seems to be significant, and there's significant quality of life improvements. So this was a small phase 1 trial. The primary purpose was to assess safety. It was not designed to assess efficacy, but clearly intriguing and promising. And based upon this, it certainly merits further study and exploration. Again, just to put a kind of a personal touch on this, there's a patient who's been a long-term friend of the Chordoma Foundation, Todd Balfe. He's an avid cyclist as well as an author. He came and visited us in the lab earlier this year, and he was one of the beneficiaries of this drug. And here you can see him on his bike. He just biked, I think, 350 miles to do a fundraiser for the foundation. So, he's subsequently gone on to another therapy, but I think just case in point that there is reason to believe that even a therapy that is not curative can buy time to enable opportunities for further advances and further therapies. I realize I'm a little bit over, so I'm going to try to go quickly here. But now I want to kind of look ahead a little bit. What's coming down the pike? What are the other big waves that we're going after? So I would call kind of the SHP2 story a small or medium, a normal size wave. There are some mega waves or mega trends that are coming in the world of oncology. And one of them is kind of this idea of surface targeting therapies. So there's 3 different modalities that, so backing up, most drugs, all the drugs we've talked, or many of the drugs we've talked about, they kind of get inside the cell and they kind of cut the accelerator or they press on the brake and stop the cell from growing or kill the cell. But there's another strategy for developing drugs against cancer, which is to try to find something on the cell's surface that could basically be a beacon where you could direct either radiation or a cytotoxic, like a chemotherapy, a poison basically, or immune cells. And so if you happen to come across these names in the news, there's a lot of discussion about them recently. So things called RLTs, radioligand therapies, that's kind of attaching radiotherapy to an antibody and having it home straight to the tumor. ADCs, that's antibody drug conjugates, again, attaching a toxic chemotherapy payload to an antibody, having it direct to the tumor. Or cell therapy. These are things like CAR Ts. It's been in the news a lot. So all of those therapies depend upon finding something on the surface of chordoma cells that differentiates chordoma from normal tissue. We don't have those yet. However, there's a number of new approaches and technologies that are making this more feasible. And so last year we invested in a project in Toronto. Some of you, I think, contributed to this project, so thank you. That project has gotten underway. We've just executed a grant agreement with University of Pennsylvania. They're going to be applying an approach to kind of tackle this problem and identify surface targets. And we're about to support, or currently raising funds to support, a project at the Broad Institute for this purpose. In the meantime, there is a kind of encouraging development that could potentially serve as an opportunity for chordoma patients. The first tumor-agnostic approval of an antibody-drug conjugate occurred this year. So it's a drug called Enhertu. And what it means is that if your tumor expresses the target of this drug, it's called HER2, then regardless of the tumor you have, the drug is available to you and approved. So the only question we have to answer is, do chordoma patients express this protein on the surface? There's some evidence in the literature that some chordoma patients do. So one of the things we're doing in the lab very quickly is trying to validate that and determine what proportion of chordoma patients, to what degree, et cetera. But this is potentially a drug that is already approved that could be applied to some portion or some subset of chordoma patients. Another thing that I would venture to say many of you have probably heard about in the news is the tremendous advances that are happening in AI and how that relates to drug discovery and development. So there's headlines in places like the New York Times saying AI is revolutionizing drug development. There's too many companies even to keep track of at this point, over 1,000 that are employing AI approaches to drug discovery and development. And we are determined to make it possible to apply those to chordoma, and that's a big part of what we're focusing on in the lab. And I guess just to briefly summarize here, the big thing, there's a lot of things that AI can do, but the big thing AI does is it dramatically reduces the difficulty of doing chemical screening. So kind of taking a large library of compounds and seeing whether they bind to a protein. Now all that can be done basically in a computer as opposed to in a wet lab. Or designing compounds, so basically the work of a medicinal chemist. So it's now kind of orders of magnitude easier, faster, less expensive to do those steps. However, what the AI comes up with still has to be tested in a biological system, and that's where the bottleneck lies. And the companies that are pursuing or developing these AI approaches, for the most part, they're computationally focused or they're chemistry focused. They're not biology focused, and they're certainly not focused on kind of chordoma laboratory capabilities. And this is where there's this kind of perfect marriage or perfect opportunity for Chordoma Foundation labs to unlock opportunities or to unlock the potential of AI to develop drugs for chordoma. Just in the interest of time, I'm not going to go into depth here, but basically just to say that this is happening now, and we're starting to initiate a number of collaborations with companies and already seeing the fruits of those efforts. So just in summary here, I think the take-home message here is that over the last 15 years or so, investments across this R&D continuum are yielding a growing pipeline of therapeutic options, some of which are now available to patients in the clinic, some of which are still in clinical trials. There are more clinical trials coming, and I think the important thing is just the message to drive home, and a big reason for hope is that what is coming next is better, more promising, with more rationale and more advanced therapies than what we've seen to date. So I think every reason to believe that as additional clinical trials read out, that the results are going to be even stronger, more promising than what we've seen recently. So what does the future look like? Trials based upon strong mechanistic and preclinical rationale as opposed to kind of simply an empiric approach. Personalized therapy as opposed to a one-size-fits-all approach. Combinations instead of single agents. New drugs instead of old drugs. So this is happening, and it's happening really because of this community and so many people rolling up their sleeves, contributing in whatever way you can. A tremendous effort on behalf of, literally hundreds of researchers across the globe, and I can't, thank all of the investigators that are working on chordoma enough. It's really extraordinary, the progress that's being made, and the number of clinicians and investigators who are dedicating themselves to this disease. I find so much hope in. So, thank you to all of you. It really means a lot. With that, I'll hold questions for the panel and invite up Dr. Walensky. I think it is safe to say that if the U.S. were to field an Olympic team of chordoma clinicians, Dr. Walensky and Dr. Gandhi would undoubtedly be on that team. Dr. Walensky is one of the world's leading spine surgeons and happens to be right here in Chicago at Northwestern. So, Dr. Walensky, without further ado.